it's not the easiest time for anyone right now. So I really appreciate your, kindness to this event. So, so this technology is ion imaging CMOS semiconductor technology, and it enables non-invasive, label-free, and real-time digital measurement of extracellular ion changes in a two-dimensional space. So, and then this new technology will get the first instrument in the U.S. At Hinge Therapeutics, my company located at Biolabs LA at Landquest Institute. So we get the first system next month. And so Hinge Therapeutics, Toyohashi University of Technology, and University of Yamanashi is there. So there's two universities in Japan are collaborating to advance technology and develop new disruptive approaches to drug discovery and also diagnostics. So this effort will be it's supported by these three entities, but for drug discovery application, Hinge Therapeutics will kind of lead this effort. So, and then we'll focus on developing applications in areas such as immuno-oncology, neurological diseases, ion channel, kidney disease, cardiovascular disease, anti-aging, inflammation, and viral infection, and which also includes COVID-19. So today's just agenda, as you know, first I'll give you a little more details of the technology, more like overview of technology, and then talk about my company and some potential application of this technology in our drug discovery effort. Second part will be by Professor Koizumi, who is expert in brain science, and to talk about how he's using this technology in his research. And then the last part will be by Professor Sawada, who is the inventor of this technology, and I'll give you more details of the mechanism of detection and the current most updated status of the technology. And then I give you just a quick intro of three speakers. I am Akiko Futamura, CEO of Hinge Therapeutics. Many of you probably know me from Infinite Bio. I still have the consulting companies called Infinite Bio. I've been running since 2006. Some of you may know me from Karna Biosciences. I was a US head for several years. My scientific expertise is actually protein chemistry, structural biology. I have PhD from, I did my undergrad at the University of Tokyo, and I got PhD from University of Illinois. And then Professor Koizumi, he's from University of Yamanashi. He's also president of Japanese Society for Neurochemistry. So he's very accomplished, very famous professor in neuroscience field. He's interested in physiology and the pathophysiology of glia to neuron interaction, and interested in ATP perinetic signaling. And so he, just to quick background, he went to Kyushu University for PhD, and since 2007, he's been a professor at Yamanashi. And more details can be found on my website, or if you need any information, I'll also follow up with everyone to get all the details. Professor Sawada, he is the inventor of this technology. He is a very accomplished, very successful professor in Japan, and of course globally he's known. And so I was intrigued by his background, but he did his undergrad and graduate studies at the Toyohashi University, and then later he went back to become a professor. So I can give you more details, but maybe just please read his bio, or I can send you more information. So the next part is actually my talk, and so the overview is, so first I give you a quick overview of the technology, and the second part I'll give you more talk about my company, Hinge Therapeutics, and what we do at what kind of drug discovery we are trying to do, and third party is how we plan to use this technology for our drug discovery effort. This is just a technology highlights. So this device, the black, this instrument is about 7 pounds, and you see that it's a relatively small. It's a it's smaller than PC next to it. And however, this is a very powerful instrument. It can do real-time level-free multi-ion measurement, and because it's level-free, it's very simple workflow. And then because it's level-free, you don't need to worry about signal weakening. In a sensor in the middle, Professor Sawada will give you much more details. There are 50,000 sensors on one chip. So this gives us flexibility in the future to potentially multiplex. Or if you want to use the entire surface, you can do a very nice spatial, phenotypic assay with spatial information. And also because there are 50,000 sensors in a small pitch, or I think I should let Professor Sawada talk about this, but we think we can do a single cell analysis. And it's a very rapid measurement is possible. It detects 2,000 frames per second. And then also, as I already described, if you use the whole surface, it can really have a nice 2D imaging with your tissue sample, for example. So this technology has been used in vitro experiment, ex vivo, and in vivo. And this slide, I just want to focus on the chip itself. The chip, this actually measures the protein concentration. By having an ion-sensitive membrane on the surface, we can look at potassium, sodium, your protein, of course, and calcium concentration. If you immobilize enzyme which generates the reaction, where the reaction generates protein, then you can look at acetylcholine or ATP and some other chemicals. So next part, I just want to give you a very quick overview of my company, Hinge Therapeutics. This was formed in 2017. Our goal is to establish a platform where we can design small molecule or small peptide molecule against many of the undruggable targets. So I give you more details about undruggable, what is undruggable. So in terms of disease, we are working on oncology, some blood disorders, CNS, and some rare disease. So we have two locations. The main location is the Torrance, that where the incubator is, and then we still have a location in Sunnyvale, in Silicon Valley. So I just want to, for people who are not too familiar, I just want to explain what's undruggable. So undruggable targets are usually difficult targets so meaning when you're designing drug, many people go after some of the easier targets, meaning so it has a very clear defined enzymatic activity with a binding pocket where the drug can bind. However, many of the proteins in your body are considered challenging or undruggable because even though they are implicated in some of the important diseases like cancer or some rare disease, these targets are considered difficult to target because of maybe they are lacking clear enzymatic activity, or is very kind of fuzzy, moves around too much, or you can't find any binding pocket where people usually try to design drug to bind. So we are going after um, difficult target. And then we also use finding the rules and find the AI to make it more efficient and potentially find novel drugs and diagnostics. So in the next slide, I just sort of summarize. So there are many AI companies, there are many companies recently targeting undruggable targets. And so, most people say, oh yeah, I know it's that kind of company, but we think our approach is very different because for several reasons. And the data we use, we usually don't care about like three-dimensional structure, or we don't care about binding pocket. Most companies look for. We care about more like mutation, how translational modification change activity, more phenotypic um, relationship with the mutation, and also we really like to know the conformational change data. And we also target, we go after very different, we look like fuzzy proteins, more difficult the better. And then our definition of good molecule is different from our conventional good molecule. Good drugs usually have a high specificity, very tight binding. For us, tight binding doesn't trigger conformational change, which we like to see. So, I mean, the type of molecule we find useful for us is usually relatively weaker binding that triggers conformational change in that context. So specificity, we think the specificity comes from the context or, the way it moves. So because we have a different criteria for our drug discovery, the validation steps are also a little bit different. And so we do a lot of biophysical validation using conformational change to monitor conformational change. But we also really need phenotypics assays because undruggable targets are difficult to analyze. So we want to see the result in the context of cell or tissue. So that's part of why we think we we have a good synergy. We wanted to work with Toyohashi to commercialize a good phenotypic screening technology. So this is just a quick overview. So next part, this is going to be the last few slides that I want to kind of focus on the technology again and see how, I want to elaborate some of the key features of the technology and how we think this can be used in a drug discovery environment. And yes, so this is going to be the last part. So I kind of emphasize it's a real time, label free, and of course multi-ion. There are many keywords that come with this technology. And label free usually means so you don't need to label the sample with fluorescence labels. You don't need to, basically have this special step to make it more to visualize it. So fluorescence detector can visualize the molecule, for example. So label-free technology, on the other hand, you don't need to do this in order to see the reaction or concentration that you're trying to determine. And so basically, there's label-free meaning rapid assay development can be also no attenuation in its signal. So usually fluorescence labels are very bright at the beginning, and then but it's gonna get destroyed with laser that you're using to visualize it. So whereas label-free technology often gets saves the sample from damaging. And let's see. But however, usually the limitations for label-free technologies, you're not looking at specific information. You're more looking at the average of certain reaction, or there are several limitations. So usually people use label-free technology and label technology together. So I think even though this is a different type of technologies, I think some of that would be applied to our situation also. So in terms of real time, technology. So because it's real time, you can look at kinetics. You can also get the result faster. You can look at shorter. The experiment can be shorter. And then in this case, we can, with this technology, we can look at the local changes, dynamics. Um, Also, because of the rapid detection, we can look at ion channels activity very effectively. In a multi-ion aspect, it's very important that even though it's level free, you can actually see many different things at the same time. So that helps increase the accuracy because you always have some control ion that you can compare. You're not just looking at one ion changes. And also that gives us possibility to do machine learning with the results to get even more accurate results. So also I wanted to emphasize a single cell analysis aspect. Um, the newest chip that Professor Sawada will talk about will enables where there are 50,000 sensors basically in one small area, and if each sensor can capture one information from one cell, you have potentially a large cell population, but you can get with data from each cell. So that's called the single cell technology, and it's very sort of almost like a trendy topic these days. Because if you look at the tumor sample or even stem cell, any samples, those are you're looking at the heterogeneous cell population, and any data you get from um, the death population is an average of what each cell is doing. So with single cell analysis, you can kind of see what percentage of the cells are doing this category. You can see like a different response from different cells. So that's very powerful approach. So we want to take advantage of that aspect. And so when we look at the, say, tumor microenvironment or some neural, some brain slice that the Professor Koizumi will talk about, we can potentially apply that to understand what the single cell aspect. And then some obvious application is also from here in the next slide. I just want to change angle, and it's when we will talk about more on application itself. So we definitely want to use this technology for ion channel studies. And of course, there are so many ion channels that are involved in very important diseases. And many current approach involves, say, for example, there are calcium sensitive dyes that are readily available, but it's looking at inside the cells, whereas we'll look at the rapid changes outside the cell. So we definitely kind of, give a new kind of information than other conventional method. Also, the technology is good for fast reading, which is very powerful for ion channel studies. And then one of the most important areas we think is the use of this technology for immuno-oncology, understanding tumor microenvironment, and potentially application for CAR-T development. Many of the immunology or oncology-related response or interactions involve very large, I would say large enough ion changes. So of course, apoptosis or immunosuppression. It's kind of interesting. New areas emerging for immunosuppression in tumor microenvironment, and ATP or adenosine concentration. That's very kind of hot topic these days. And then T cell regulation, T cell interaction with tumors, T cell activation status. It's all involved. All the ion changes are involved in this process. So this is my last slide. So summary and future directions. So this new system will be installed early July, so next month at our location. And so, if you are visiting, if you're in the same incubator, please definitely take a look. And then we want to explore new applications in oncology, immunology, CNS, and diseases resulted from ion channel dysregulation or dysfunction. We want to explore possibility of single cell analysis capability and apply it to especially in immuno-oncology, especially including CAR-T research. We will now start working on the machine learning aspect of this multi-ion results. And then we actively look for partners potentially for each application. We'll do many by ourselves, but we love to work with other companies. And any input will be always welcome. And the goal is, ultimately, it's really to advance the science and save lives. So that's the end of my talk.
The recent trend in brain sciences and biology is from macro to more and more microscopic direction. The study of humans and organisms are roughly divided into some stages or hierarchies, that is, whole bodies, organs such as brain, heart, intestines, cells, and molecules such as genes. Of course, these stages are very important, but scientists are working in a more microscopic direction, this direction. Also, scientists tend to work on more and more about intracellular molecules. For example, these receptors and the channels in the plasma membrane. And scientists are more interested in these receptor-mediated intracellular molecules such as calcium ion, IP3, and so on. And thus, now we knew huge number of receptors and intracellular molecules, cascades, and their interactions or crosstalks like this. However, brain function is not such simple. That is achieved through intercellular communications of various cells. That is not understood only by any detailed analysis of intracellular signals in a single cell. Thus, imaging of intercellular mediators between cells is essential, which requires a high-resolution spatial-temporal analysis of extracellular microspaces, that is niches. It also requires simultaneous imaging of multi-molecules at the same time. As for the recent topic of brain sciences, they have shifted from neurocentrism to neurogliology. This is a classical view of synaptic transmission. Synaptic transmission was considered to be mediated by neuron-to-neuron communication like this. However, if we look at synapses by electron microscopy, we can see pre- and post-synaptic structure is surrounded by this red signal, that is glial cells, astrocytes. And the astrocytes look as if they form the third synapse. Actually, they communicate each other by using multiple molecules via these very complex niches. This is the latest view of synaptic transmission and tripartite synapses. This is consists of neuronal pre and post synapses, as well as glial perisynapses. This is a more detailed example. When neurons 1 and 2 are excited, they activate not only post synapses, but also glial perisynapses like this. And then perisynaptic glial cells release different molecules such as lactate and ATP with distinct kinetics, thereby leading to activation and inhibition of adjacent neurons respectively. Thus, multiple and higher spatiotemporal imaging and niches is required. But so far, the scientists has tried this imaging simply because there was no good equipment to analyze this. Now, the CMOS ion bioimaging sensor can achieve all of this. This movie shows experimental view of extracellular calcium imaging in the hippocampus slices using the CMOS sensor. Firstly, we place the calcium sensor chip in the device, just like this. The hippocampus slice was taken from the brain and was simply placed on the CMOS sensor with the running buffer without any labeling. The slice was stimulated by physiological glutamate, and upon stimulation, we can see the spatial-temporal dynamics of extracellular calcium signals in the hippocampal niches. It's like this. This slide shows much detailed imaging of extracellular calcium signals in the hippocampus. This is a sensor chip on which the hippocampal slice was placed. And this is the expanded image of this slice. When stimulated with glutamate, extracellular calcium in the hippocampal niches was dramatically reduced like this, which were not uniform and different in intensity and time course from place to place. These were mimicked by NMDA stimulation, just like this, suggesting the involvement of NMDA receptor channels in the responses. So far, little researchers are interested in extracellular calcium in the field of brain sciences. But importantly, such calcium decrease or changes in niches dramatically affect brain functions. When we understand organs such as the brain, we need to understand communication between not only neurons, but also many different types of cells from the brain or other organs. They communicate with each other with multiple molecules with distinct kinetics. The imaging of bioactive molecules and ions in multicellular niches has received great attention in the field of brain sciences and biology. The CMOS ion bioimaging sensor makes these things possible and could be a device that causes paradigm shift in the field of brain sciences. David.
My name is Kazuaki Sawada, Toyohashi University of Technology. So I have been investigated CMOS-based ion image sensor devices, which can be take up ion or neurotransmitter.
音声が消えてます。
This optical microscope cannot catch.
何ですか？
大丈夫です。はい、続けてください。
大丈夫ですか？
音声が消えてました。はい。
Activity.
OK。
If we can fabricate CMOS-based this like this figure, which can we can catch chemical activity directly using the CMOS devices. Then this device can be fabricated, integrate the conventional biosensor devices with LSI technologies. As you know, the image sensor is widely used in various handphone or several cameras, and the optical sensor with the highest density and defined process. So as you can see, the pixel pitch of most recent image sensor device is 1.0 micrometer. It means it is smaller than the synapses. So it means by using the CMOS image sensor technology, it might be catch the cell activity. This photograph is fabricated ion image sensor devices. The number of the pixel is 128 by 128. The pixel size is about 23 micrometer squares. This fabricated devices is operated by the LSI technologies. This blue area is sensing devices. Then this areas is selectively catch the hydrogen, proton ions. And let you show the typical images of the hydrogen distribution. Firstly, the pH 9 solution is covered on a sensor chip, and I dropped pH 6.89 solutions. Please watch these movies. By using pipette, we dropped the pH 7 solution. The pH 7 solution is distributed, and we catch the pH distribution in movement image directly by our sensor devices. This sensor can be operated by the 50 frames per second, so it means time resolution 20 milliseconds. Recently, we have fabricated more highly sensitive and more high-speed ion image sensor devices like that. This sensor, the pixel pitch is about 2 micrometer, and frame rate is 2000 frames per second. The time resolution is 0.5 milliseconds. It is very useful to understand the cell activity through the ion channels. This movie is very small, about 4 micrometer diameter crystal is dropped and dissolved in a solutions. And this is a benchmark of our sensor devices. So our work is placed on this here. The pixel pitch is 2 micrometer, and pH resolution is about 0.01 pH. So our sensor devices is much smaller, much high spatial resolution, and more highly pH sensitivity other than other work. This is also the benchmark of other devices. Recently, the TSMC and University of Illinois group presented the very 1024 by 1024 by devices. However, our sensor is more highly density, spatial resolution and time resolution. And this is another typical movies. So as you can see, very, very smooth and very beautiful picture can be taken our sensor devices. So let me show you several examples taken by our sensors. So as you know, acetylcholine is very important neurotransmitters. But however, there is no good fluorescence level for acetylcholine imaging previously. So we tried to the acetylcholine movement or distribution taken by these sensor devices. Acetylcholine, as you know, acetylcholine is very useful information for developing the medicine of Alzheimer's disease. To take the acetylcholine distribution, we apply the enzyme reaction. So acetylcholine esterase is immobilized on a sensor chip by using polyion complex membrane. And if the acetylcholine is coming by the enzyme reaction, the hydrogen ion is generated on the surface of the sensor chip. By taking this hydrogen ion distribution, it means we understand acetylcholine's existence. This is a typical picture dropped on acetylcholine. When we drop the acetylcholine on the sensor chip, so we can catch the acetylcholine distribution like that. For the negative control, there is no acetylcholine esterase on the sensor chip. There is no variation or movement cannot be observed the movement of the cannot be observed sensor chip. So we try to catch the acetylcholine emission from the cortical slice. As you can see, we put as a cortical slice is placed on a sensor chip like that, and we put the glutamate stimulations in cortical slice. As you can see, acetylcholine emission can be observed. This red areas is acetylcholine emission areas. I think this is the first report of the acetylcholine emission from the cortical slice directly by our sensor devices. And not only the hydrogen ion, potassium or another kind of ion, for example, sodium and calcium ion can be catched by changing the selective membrane. The selective membrane is then fabricated, realized by the PVC membrane and ion hole. And this is a typical example for the potassium sensing membrane. As you can see, potassium sensitivity is 50 millivolt per decade, and our outside of the cell, extracellular cell of the potassium ion is several millimolar. So it means it is possible to take the ion channel reactions. So we put the hippocampal slice on a sensor chip, and we catch the potassium emission. We try to catch the potassium emission from the cortical hippocampal slice. So this is a hippocampal, so optical microscope image of hippocampal slice and the potassium ion image taken by our sensor chip. So we detected potassium. We can successfully detect the potassium in extracellular potassium image from the hippocampal slice without label. This is a movie from now the glutamate stimulation was done. The potassium emission is occurred from the hippocampal slice. So I will move. I will show you once more. this area is CA3 areas, and this area is CA1 areas. By the emission CA3 from the CA3 areas, potassium is emitted rapidly and gradually decrease. Then we can we have fabricated another kind of ion image sensor devices. So this is a needle type ion image sensor devices. This needle type ion image sensor devices can be inserted in animals' brain. The thickness of the sensor is about 50 micrometer to 100 micrometer. So it is possible to insert the sensor devices, and pixel pitch is 32 by 128. The pixel pitch is about 30 micrometers. So this sensor is how the result taken by our sensor devices presented in Nature Communications in these years. So I insert the sensor chip like that. So this is in the brain areas, the part of the cortical slice. There is this pH distribution can be observed like that. The difference of the pH is about 0.4. I think this, and also this is a very special first observation of detection, this image distribution of the pH in the cortical slice. Finally, so this is my collaborator, who is the former president of our university, and he is a very famous biologist in Japan. He had a responsibility of Human Gene Project and head of Japan. He often said, history of progress of life science is history of advances in measurement technology. It means to the progress of the life science, it is necessary to the advance of the measurement technology. So I think our ion image sensor device to be a good measurement system for the biological field. Thank you very much for your attention.
Okay, so here we finished 3 talks, and I want to have some Q&A, or just, some, if you have any comments, we'd like to hear. And also, this is opportunity. So Professor Sawada is actually participating here, and also we have several experts actually online right now. So if you have any questions, this is actually a great time to do so. So please unmute yourself if you have any questions.
Well, I'm just very interested in the CMOS sensor being applied to biology, and I also learned something new, that the synaptic communication requires multiple different molecules, even with calcium. So very interesting. Thank you for the session.
Yeah, thank you for your comment. Mr. Okada, I've known him for several years. He is just very knowledgeable in many areas, but I think your background is more like a battery. Can you tell us more about your background?
Yeah, I'm electrical engineer. So my background is cell phones, and now it's EV charging. So it's a different field, but when I hear about CMOS, the CMOS sensor, I have a friend who invented CMOS sensor in Japan, Mr. Teranishi, so I'm very curious about that technology.
Yeah, okay, thank you. So this is so this CMOS itself, I have a few friends who are involved in CMOS, imaging but this is definitely our sort of our first time to try to integrate this more engineering side to drug discovery. But in my past experience, way before I started my company, I worked for Sumitomo Corporation's life science group, and we worked with many instrumentation companies, and we did a lot of comparison, different detection system, analysis method, and so that's why I feel kind of really excited about actually taking a different, so CMOS, it's totally different technology for us to think about new applications. And so anyway, I'll appreciate your feedback, Okada-san. And Joanne, did you hear you speak? I see you are unmuted. But yeah.
Can you hear me now?
Yes,
Yes.
I can hear you.
Okay,
Yeah.
finally. I exactly did the same, but it was not working. I don't know why.
Oh, I see. What did you think? This is the first time to hear from the professors, and what did you have any comment, questions?
It's very interesting. I just have maybe can you check? Can you study any type of ions, or is it specific to some specific ions, I would say?
Yeah, can anybody answer? I mean, like from Japan. My understanding is, but it's all kind of muted. So my understanding is you need those ion selective membrane. So some ions they are not. For example, like chloride ion, for example, it's hard to find a good ion selective membrane. So that's sort of a limit. And okay, let me tell you that also in a biological sample, if there are many similar ions, then it's harder, right? So potassium, sodium, protein, calcium, there are already like really good membranes that are also in a biological sample. Those ions are very different from other ions. So you can have a good detection. So it's a membrane and also biology sample that kind of limit what we can detect. But I think so far calcium, if you can detect calcium, sodium, potassium, and then hydrogen, then I think it's a really good set for biology.
Yeah, they are the major ions study.
Right.
I remember I did a bit of electrophysiology 20 years ago. far long time ago.
Yeah, I'm learning everything too, so yeah.
And you guys are thinking to isolate the cells from the chip? I mean, from the surface after you study them? Or
yes.
you want to study you want to study the variation of the ions?
Right, that's a good point, because no, this technology doesn't damage the cell. It'd be nice to be able to.
Yeah.
Right, so you have a good point. I think those details we'll start working on and see, if we can, what's possible, what's not. But I would think we can take the cells off and then do further, it could be, just like these damaging technique that we can potentially use. Yeah, so that's a good point.
It might be a bit difficult for tissues.
Right.
Yeah.
Okay, so I'll follow up with everyone with a presentation material, and we plan to make this presentation available either through website, or so we'll think about some logistics, but I'd like to share all this information with you. And also there are some really good publications by both professors, and then starting next month, we'll start generating data in LA at our incubator also. So we'll we hope to update everyone with newest information regularly. Okay, so I really apologize for the technical problem that I couldn't figure out the mute and my mute, but next time I'll be better. So, and then Takashima Sensei, do you have any final comments?
No, I don't have,
Anything?
but I think is it Sawada Sensei? Can you give some final some message?
Yeah, since Professor Sawada.
Okay. Oh, this is Sawada. Thank you very much to join the webinar. So now our sensor is developing. So the pixel size less than 2 micrometer. Now I have developed 1 micrometer resolution is possible to image the ion. So I have several kinds of challenge to observe the single cell activities. So it is on unpublished data, we can find the cell communications on the sensor chips. And these and also potassium, sodium, and hydrogen ions is simultaneously observed. So, for example, it is unpublished, but for example, on a hippocampal slice, on the glutamate stimulation, potassium absorption and potassium emission and hydrogen absorption is observed simultaneously, and these ion movement is occurred simultaneously. It is very exciting data for the my collaborator Koizumi. So if you want to try our sense system, please contact Dr. Futamura Akiko. Thank you very much.
Okay, thank you for the comment. And I mean, yeah, I'm so glad. I wasn't sure, any of the people from Japan can participate, so I didn't force anybody, and I really appreciate, getting up so early and be part of this webinar. Okay, I think it's kind of getting late, so I'll wrap this up. But really, I really thank you everyone for participating, and we want to do this more often, but maybe next time it's one speaker, shorter presentation, or we'll figure out the format. And also in terms of the time, like this is we did it started 1 p.m. West Coast time, but next time we will maybe we can change it to different times so more people can participate and things like that. So please give me some feedback if you have, and any questions from this webinar, let me know. And really, I thank you so much. And for Japan, have a good good day today. And some people, I think, most of the rest are West Coast, I think. Anyway, thank you so much.